So today we're going to be presenting about our mini project. Next. So history or contents, introduction and hypothesis, process, result, and references. So basically our problem when we come from project 17 where a biochemist is interested in gene copy number of kinase enzymes and the importance in terms of its molecular function. So my hypothesis is that some kinase will have a low copy number because a single gene is enough to perform their essential work, but some kinases might have many duplicate because they need to mutate and adapt to different environments. So our process will start from gathering data from NCBI and then finding kinases by using the computer command prompt. And then we use the LEVIO and MAP-T to do the sequence alignment and build the phylogenetic tree by using RAXML. Lastly, we visualize it by using Fig tree. So on step 1, we have to go to biological database of NCBI and search bacteria, Escherichia coli, and Mycobacterium tuberculosis. We then download the proteomes and the complete translated protein by using the .FAA fast cell files.
So let's talk about second step, finding the kinase. So this one we use command prompt. So basically we just log in through SSH-COM-by-067 and then we go to working directory because we already created a folder inside FileZilla and we already uploaded the proteome of Mycobacterium and also the E. coli. So basically we just cd inside that directory. Second, we filter by using GREP, which is search tool. As you can see in left picture, we use GREP for kinase in E. coli. And as you can see, there's several kinase enzymes. And then when we look at the right picture, we use GREP with the VC, which is word count, to count how many kinase. There are 130 in kinase E. coli, and then there's 70 in kinase in Mycobacterium. But if we just pick one, which is histidine kinase, that's 27 in E. coli and 6 in Mycobacterium. But when we turn to pyruvate kinase, there's only 1 in Mycobacterium and 2 in pyruvate kinase. And the third one, we use the WK to keep only histidine kinase, and then we save as another file named myco_hist_kinase.faa. So let's talk about the last one. We use CAG command, which show everything inside that file. From the previous one, we save as myco_hist_kinase.faa, right? And then we're just reading the proteome inside. For example, the first one is 2-component sensor histidine in Mycobacterium. That's a whole proteome there.
Okay, so for this step-based sequence alignments, we use MAP7 website to analyze the sequence that we get from the previous step. So, and then we open the LEVIO program to see the result of our alignment sequence to generate the phylogenetic tree in the next step.
So let's talk about building the phylogenetic tree by RAXML. So basically, the first one, we're running the model test to find the perfect match for our protein. It was the LG plus GF plus F. And then we setting to matrix, which is LG from the model test. The LG is a specific scoring matrix named after Lay and Gaskell. And then third one, we route a chain run type to ML plus rapid bootstrap of 100. ML is maximum likelihood to find a tree that mathematically makes sense. And then we use the bootstrap, mean the program rebuild the tree 100 times. This is step 5. This is the result putting in Fig tree. This is 100 bootstrap that shows the correlations between E. coli and Mycobacterium. And then we change to 1,000 bootstrap later, but it consuming more time. The 100 bootstrap take around 2 hours, and then 1,000 bootstrap of histidine alone was taking around 11 hours.
Okay, so for the last one is our result of mini projects. We found that a huge difference in the copy numbers between the two species. In the E. coli, had a high number of copies with about the 27 copy. These sensor genes, in contrast, Mycobacterium tuberculosis have a very low copy number with only 6 copy. When we looking at the phylogenetic trees, we can see pre-aligned age-specific explanations in the E. coli in the simple term. The gene in the E. coli copy themselves many times, forming tight group called interlock. All of these cluster genes in the E. coli act as like a co-ortholog when compared to the few's original genes found in the Mycobacterium tuberculosis.
This is the concept structure result. As you can see, this is a primal result. When we look at the pink one, which is Mycobacterium, and the blue one is E. coli, because the kinase must maintain a 3D shape to do its job. Although the evolutions amino acid can mutate it, as you can see in UniProt, the percent identity matrix, although it's the same species or different species, the percent identity is quite low. But when we looked at the 3D structure in primal, you can see there's nearly superimposed to each other because as long as the mutations doesn't destroy the final folded 3D shapes, the bacteria can survive.
Okay, so this slide I will talk about why are there so many genes copied in the same species. The answer is very simple. It's about the adaptations. In case of E. coli, it live in the several environment like in the water, in soil, or in the human gut. To survive, it use the gene duplications. The original genes keep doing its essential work, while the new duplicated genes are free to mutate and adapt to detect the different environment. So in contrast, Mycobacterium tuberculosis live in a very stable environment inside our lung. It doesn't need to constantly adapt, so a single genes or a very low copy number is enough to perform its essential work. It simply doesn't need to waste the energy making an extra copy. So the last one is our limitations of my work. We have two main points. The first is our work is computer predictions, or even though we see these copy genes on the tree, we cannot prove that it is happen in the real life or not. We choose to have a lab like a real-time PCR in RFI to see this is happen in real life or not. So the second is we face about the computer limitations. When I work, first we have this 200 sequence to analyze of kinase genes, but it takes over 9 days to complete it. That's why we focus this only in the histidine kinase to analyze the genes both of the organisms. And this is our reference of my project. And thank you.
Okay, first go back to the conclusion slide that you talk about the limitations.
Limitation.
So you said that analyzing 33 histidine kinase gene took 11 hours.
Yes. Yes.
So are you analyzing on your laptop?
Yes, our own laptop. Yes.
But I think in the class, I already give you or demonstrate you the options that you can run the phylogenetic tree on server. So this shouldn't be the limitation.
I didn't do it on server, just normal applications.
Okay, so what I want to say is this is actually not a limitation, and actually you can run on the servers, and it will take much less time because I have experience running much more like larger data. These are only 33 genes, right, from 2 species.
Yes.
So just only 33 sequence. And how long is the sequence? How long is the alignment?
The alignment or the building the tree alignment to
the length?
very few minutes or few seconds.
No, I'm asking about the length of the sequence alignment.
Oh, length of sequence.
So you do amino acid sequence alignment, right?
Yes.
Have you take a look at the alignment result before you're going to perform phylogenetic analysis?
Just in this
I guess it wouldn't be.
this one in the picture, I will look at it, but I haven't looked the total length, but I look at if they really alignment.
Okay. I guess it shouldn't be that long. I mean, it shouldn't be very long, so it shouldn't take very long time to run the phylogenetic analysis. Okay, you already mentioned that you open the alignment on allele and you try to check whether the alignment is correct or not. What are the
Yes.
criteria you use to check whether the alignment is correct?
I look at the before alignment and after alignment, and I try to compare with different alignments too. Alignment on this allele, and I've changed to MAPT in left picture too.
What are the criteria?
What are the
Alignment
criteria?
is not the alignment. Allele is not the alignment tool, actually. You can perform well, but there is a function in allele that allow you to perform alignment. And I guess the aligner inside allele is probably muscle, which use quite similar strategy as MAPT. So if you actually done the alignment on allele and then compare with MAPT, you probably didn't get much difference. Okay. So then what is the criteria you use to decide whether the alignment is correct or not?
We didn't have the criteria to select, but we select the one on MAPT to analyze. Yeah.
Okay, why don't you use other program like Cobalt that probably help you to identify the domain, or try to like arrange the alignment to identify domains somehow. Why don't you use that program?
Because we just know the step that before going to RxML, they need just sequence alignment, and then we have the data, and then we jump directly to the RxML.
No. I'm asking you, why don't you use Cobalt instead of MAPT?
I have no idea.
Okay. Why you select these two bacteria?
Bacteria.
Yeah, you because I think in the project, อาจารย์ Thitiphan didn't. I mean, he didn't specify the organism's name, right?
Yes.
He allow you to choose whatever organisms you want to work on, and you decided to work on only 2 species.
Yes.
Why you select E. coli and Mycobacterium tuberculosis?
Okay, so for the first plan of our project, I used the E. coli and compare with the bacillus. But our result is gonna be like, we found that the copy number is only 1, but I cannot prove that it is correct or not. That's why I changed it to Mycobacterium, like E. coli and Mycobacterium, to find the histidine kinase, because this proteome its function is about to find food. I think it's quite easy to find the copy number of genes. That's why I use this.
But then why it has, well, for E. coli, it's I kind of see the reasons, because you might familiar with E. coli. But why you have to compare with Mycobacterium? Why don't be other type of bacteria? Are they the same gram positive or negative, or what?
ตอนแรกตรงนี้เป็น positive ผมเปลี่ยนแปลงแต่
เขาเรียกว่าศิลาแต่คุณเรียกเป็น negative
Um, the first try, which had mentioned, we select gram positive to compare with gram negative, but when the result come out, it just quite quite confused, and then I've I've tried to search another one, and I tried to pick from the environment instead, because the first try, the gram negative and gram positive, I cannot like clearly distinguish from each other. But when I look at the environment, the E. coli is like from the nature, but the Mycobacterium tuberculosis live inside our lung, and the histidine kinase show very different from each other. So this is why I selected. It's come from just thinking of where they live and pick it.
Actually, it's hard to compare. If you choose other bacteria that live in the gut or probably have some common environment with E. coli, that would be better, I think. And why did you select histidine kinase? There are multiple types of kinase, right?
Yes.
Why you decided to analyze histidine kinase?
The first try was the pyruvate kinase, and there's two in E. coli, and there's only ones in Mycobacterium. But when we analyze and we see in FICKI, it is like a three different separate line, and then I'm quite confused because it's very low number, cannot like tell that it like a paralog or autolog, doesn't like see that difference. But I tried to like randomly pick another one that I filter only kinase, right? And then I tried to look through all 130, and I found like histidine kinase quite a lot. But when I look at in Mycobacterium, the histidine kinase was like very low. So I tried to find like a correlations, or like why it's like is see a lot in like the first row that happened when I filter only kinase. So I just picked it, and then I just use word count, and then it's like if number is like show like not too much but not too low like pyruvate kinase, it would be better to see the result.
It seems that you guys, instead of using biological knowledge or any scientific reasons, like the first group I already give the same comment, and this one I'm going to give the same comment that you just choose the one that you can manage it, right?
Yes.
Well, not a bad idea for the relatively short time, but that is not a good idea in science. You get my point?
Yeah,
Okay.
yeah, I get it.
And you should have a summary table to present the frequency of each of different type of kinase that will help supporting the reasons of choosing histidine kinase. I don't see that table. Can you show me the phylogeny? I cannot clearly see the phylogeny. Okay. Why there are two phylogeny here? The first one.
The first one was 100 bootstrap, and the second one was 1,000 bootstrap.
One thing that I have one thing that I have to emphasize here is that the lowest number of bootstrap replicate should be 500. So if you do 100, it's just for demonstration, or if you want to see something real quick, but you cannot report that. 500 bootstrap is the lowest one. But normally, you should go for 1,000 bootstraps at first. So I'm not going to consider the 100 bootstraps phylogeny. Okay, well, let's take a look at 1,000 bootstrap. And have you wonder about the bootstrap value because I
Yes.
see it's relatively low. Only
Yes.
a few clade that show acceptable bootstrap support. Acceptable means higher than 50.
Yes.
Okay.
Um.
Have you wondered about why it shows something like this?
I've tried to like think why, and I try to think of different reasons, but one reason I think is like when we look at the, for example, this sequence alignment, there's one line that doesn't like align with the other one, is take the other one to have too many dash, and then when too many dash, it's like the program have to like guess too much whether that is belong together. It's like a missing data, I think. So it's like it's kind of low for this reason, I think.
Maybe, but I don't think. Well, if you take a look at the name, go back to the phylogeny, the letter is very vague. It's very small for me. So they are just simply histidine kinase,
Yes.
sensor histidine kinase, two component sensor histidine kinase. You also have putative two component sensor histidine kinase. So basically, let's say we just include the putative to the one that has that doesn't have the term putative. So basically, you have three groups, right? One is histidine kinase, another one is sensor histidine kinase, and the last one is the two component sensor histidine kinase.
Yes.
Have you checked whether these kinase have different domain compositions?
I haven't checked about domain.
Okay. So have you take a look at so you didn't perform any domain analysis, right?
Yes.
Have you take a look at the annotation of each sequence? At
Yeah,
least like just take a look and see the position of the kinase domain or the number of kinase domain in this three types of
I doesn't look at the annotations.
Okay. So that would help you interpret your alignment, and that would help you decide whether you're going to exclude some part of the alignment to make it more, like make alignment look more consistent, so more, how can I say, or help increasing the bootstrap support on the tree. So what is your interpretation of your phylogenetic tree? So you said that, okay, I think you list down here. Show clear lineage specific expansion. Could you point out on your tree, on the phylogenetic tree that support this statement, the clear lineage specific expansion?
Expansion.
ย้อนกลับไปหน้า
These three, yeah. So
เดี๋ยวประมาณ100ครับนี่เดี๋ยว
Maybe this one, there's 100% in E. coli.
that's all?
Or this one too. This the bootstrap is 100 in E. coli too.
But those are just two smallest clades. I mean, what is the meaning of clear lineage expansion? So clear lineage specific means that you expect to see the monophyletic clade of the kinase of histidine kinase, that all of them are belonging to one species. Am I correct?
Yes, I think.
Have you seen that on the phylogeny? Did you see anything like that on your phylogeny? It has to be big, right? Because you have many kinase, many histidine kinase in E. coli and several histidine kinase in Mycobacterium.
เนี่ยอันนี้ตัวเดียวเลย
ตัวเดียว
ลายเดียวเลยใช่ครับ
The last line.
Whoa.
The
So I don't agree with your conclusion. Because if you're going to show the lineage specific expansion, go back. I'm afraid I will use the wrong word. Yeah. Lineage specific expansion. What I would expect to see is two big monophyletic clades. One presenting all histidine kinase in E. coli, and another one present all histidine kinase in Mycobacterium. Did you see that pattern? I don't think we see that pattern, right? Even though we see that pattern, the bootstrap support is still the problem because the bootstrap support, the large monophyletic clades is relatively low, right? Just 1, 2, or 11 is relatively low. So I don't think that the first conclusion is correct. So let's start. Okay, how can you identify co-autolog? Yeah. You said that these clusters act as co-autolog to the fever ancestral-like kinase gene in M. tuberculosis. What is the meaning of this statement actually? Could you explain?
Because-
Well, can you explain in Thai? I just want to make sure that I understand it correctly.
โอเคครับ ก็คือในอีโคไลมีฮิสทีไคเนสใช่ไหมครับ แล้วก็มันมีวิวัฒนาการมาต่อเนื่องเป็นแยก copy number ออกมาเป็นแล้วแต่ฮิสทีไคเนสแล้วแต่ environment ของแต่ละอีโคไลนั้นๆนะครับแต่ว่ามันก็มีอีกตัวหนึ่งที่มีวิวัฒนาการร่วมกันก็คือมาจาก Mycobacterium tuberculosis ก็จะเรียกว่าการวิวัฒนาการเรียกว่า co-autolog ครับก็คือมันมาจากบรรพบุรุษเดียวกันแต่ว่าแยกสปีชีส์กันนะครับจะเรียกว่า co-autolog.
Okay. Can you point out the clade that's supporting this statement?
Oh my god.
ช่วยลองเนี่ยตรงนี้ครับมีคนลายกับมันคือเรื่องปุ๊บแล้วก็ไปจุดที่คุณปุ๊บมาเนี่ยแล้วก็ปุ๊บแล้วก็อันนี้อีโคไลอันนี้อีโคแบคทีเรียม
So you mean that the two component sensor kinase in Mycobacterium has common ancestor with the sensor histidine kinase
ตัวไหนเชื่อมกันระหว่างสอง
เนี่ย
Mycobacterium
82เลย
82ต้อง80
แล้วก็บูสตแรปที่ลงมานะมัน88 100 อันนี้ไมโครแบคทีเรียมในอีโคไล
มันอยู่
but then well, I think I see. So the monophyletic clade that support with 82% bootstrap support, right? And I see, and the upper two are two component sensor kinase, histidine kinase present in Mycobacterium. And the other three are the sensor histidine kinase in E. coli. Okay. Do you have the full names for these three sequence? Because I see something weird here.
full name
Yeah.
คือ
So they are all from K-12, different NP accession number. But okay. They'll explain. You know what UHPB and ARQ and ARH are? Okay, last question because I think we ran out of time. How did you collect this sequence? Did you collect the protein sequence from a single record of E. coli? Can you go back to the material method section that mentioned about the sequence collections? So search bacteria, E. coli, Escherichia coli. So, okay, from genome sequence.
Yes.
Okay, so you use so you download the protein sequence from this genome assembly.
Yes. Yes.
Okay, makes sense. Okay. Well, even though it's show the whole autolog, but the pattern was not. It just like just part of the phylogenetic tree. Do you see my point? It's well, I cannot say it wasn't support, but I still don't feel comfortable to draw that conclusion because you just see the part of it. I mean, and you didn't do any further discussion on those specifically on those claims. คือคุณสรุปคือข้อสรุป3 ข้อที่คุณสรุปมานะมันไม่ใช่แพทเทิร์นที่แบบสามารถสรุปได้ทั้งทรีมันเหมือนแบบหย่อมนี้ใช้ conclusion นี้ได้หย่อมนี้ใช้ได้อะไรเงี้ยเข้าใจไหมมันไม่ค่อยสตรองเท่าไหร่ okay. That's the point. Okay, that would be all for your group. And I would one comment that I think you should revise before you submit your summary is the picture. Well, but I think if you can increase the text size on your presentation. No, I'm trying to think whether I allow you to submit any file on the Google form, but I don't think I have that for you. So maybe try to find any strong conclusions that explain your phylogenetic tree, okay? And then put it in your summary. Okay, that would be all.
ครับขอบคุณครับ
I think I see อาจารย์อรุณทิพย์